Hey guys. So we have been looking at organic molecules this week as part of our biochemistry unit. Um, and we've already looked at enzymes, we've looked at carbohydrates, we've looked at lipids. The last one that we have left is proteins. And I know that enzymes are proteins, and nucleic acids we'll get to in a different unit. And so we're going to take a look today at the structure of proteins. Before we do, I want to start with a bell ringer. So go ahead, pause, and answer this bell ringer in the bell ringer section of your binder. Remember to write the question and the answer and the date. So hopefully you know that the organelle that controls the cell's activities is a nucleus, and the organelle that makes proteins is the ribosomes. And so since we're talking about proteins, proteins are made by the ribosomes. We will actually get into protein synthesis later this week, but there's to help you with the bell ringer if you need it. So we're going to go ahead and take a look at this color sheet. It's one of the ones that I posted on September 28th. I hope you have it printed out and ready to go. If not, you need to pause and get that taken care of so you can finish this. Let's go ahead and begin. It says, organic molecules provide the body with structural materials to form cells, tissues, and organs, regulatory substances to direct and govern the interactions of the molecules, and energy to fuel the chemical operations of the cells. This is just some of the jobs of the organic molecules. They are building blocks, that's the structural components. They help to govern the interactions of some of the other molecules, and they're energy sources, right? It goes on to say in the previous plates, we discussed the structure and function of the carbohydrates and lipids. Here we're going to look at proteins. And what I want to highlight or underline first is kind of this next sentence. It says, proteins are vital to the formation of, and function of many cellular structures and processes. They are also among the most diverse organic molecules in the living organism. There are lots of different types of proteins. That's what diverse means, right? Lots of variety. We've already seen integral proteins, alpha helix proteins, glycoproteins, and peripheral proteins that are all in the cell membrane, and they have different jobs in terms of helping to form the shape of the cell or moving things in and out of the cell, or even the cell identity and holding on to those carbohydrate chains. We've also looked at enzymes, which are proteins, and the idea that since every enzyme can only catalyze one reaction, that there are thousands of different types of enzymes, right? And that hasn't even really begun to look at what are some of the other proteins that form your muscles and various other organs in your body. And so they are an extremely diverse group, right? Goes on to say here, this plate shows how amino acid subunits are joined together to form protein molecules. Once formed, proteins can assume a number of shapes. So let's go ahead and highlight or underline where it says amino acid subunits are joined to form protein molecules, and once formed, they can assume a number of different shapes. And so you kind of saw a little bit of an intro to this in the Amoeba Sisters video that you watched, that proteins are long chains of these things called amino acids. In the Amoeba Sisters video, it mentioned the monomers are just the building blocks of some big chemical. So like in carbohydrates, the monomer was monosaccharides. In proteins, the monomer is amino acids. Proteins are made out of chains of amino acids. And that's kind of where they go next. Proteins are molecules that are formed from units called amino acids. A protein may contain as few as 10 amino acids, or it may contain thousands. The sequence of amino acids in a protein gives them unique functional characteristics. There are, it's like 20 to 30, I don't remember the exact number, different amino acids, and there's so many different ways that you can combine them. So that's what they're getting at with the sequence. The way that they're combined or connected kind of determines what's its job going to be, and how does it work. So let's go ahead and continue on. Make sure that you are pausing and highlighting or underlining with me. And so pause and go back if you need to, highlight or underline together with me. In this plate, we will show how amino acids combine with one another to form a linear protein called a peptide. So we're going to highlight or underline amino acids combine with one another to form a linear protein called a peptide. Linear just means that it's in a line. That's a math term. So it's a long line. It's not like that spiral chain where we saw with the plant starch. This one is linear. It's a line. And it's called a peptide. Upper portion of the plate shows 2 amino acids outlined by brackets. Each amino acid contains a carboxyl group and it says shaded on the left, amino acid, and outlined on the right one. They're talking about in the diagram on the other side. Amino acids also contain amino groups. It says in the left, the amino group is outlined, in the right one, it's been shaded. So again, they're just explaining on the diagram and coloring instructions. The amino group contains nitrogen and hydrogen atoms. And then there is also an alkyl group. The R stands for a general alkyl group. And each of the 20 amino acids bear a distinct alkyl side chain. So this is getting at the structure, which we'll look at in a second on the back. But essentially, here's what you need to know from this paragraph right here. I'm going to turn it sideways, and you can write this on the side for yours as well. Amino acids have 3 parts. So again, a protein is a chain of amino acids, so we ought to look at what is an amino acid. If a protein is a chain of these, then what is an amino acid? So 3 parts. There is a carboxyl group, an amino group, and an alkyl group with a side chain. So pause and copy that if you need to. These are the 3 parts of an amino acid: carboxyl group, an amino group, and an alkyl group with a side chain, right? And so this is what is an amino acid. And then proteins, of course, are chains of amino acids called peptides. All right, so just recapping. Proteins are chains, big long chains of these monomers, these building blocks called amino acids. A chain of amino acids is called a peptide. We're going to look at a linear peptide first, but they can have other shapes. So if a protein is a chain of amino acids, then what is amino acid? An amino acid is a molecule, an organic molecule that has 3 basic parts: carboxyl group, the amino group, and the alkyl side chain. If you were in my physical presence classes, I would have you try to guess what is a carboxyl group. If you think about it, we've heard of hydroxyls, and you should know what a hydroxyl is with its hydrogen and oxygen. So what's a carboxyl? Let's go ahead and take a look at these. So as you flip over on this side here, it's going to show you these 3 parts, okay? So the first part that it mentions is an amino group, and that's this part right here. This is an amino group. You can pick 3 pale colors. You're going to need 3 colors. This one's the amino group, right? And all amino acids have an amino group. An amino group is a nitrogen with either 2 or 3 hydrogens attached to it. It's kind of interesting because we've been learning that atoms have to have a specific number of attachments. So how is it that sometimes nitrogen can have 2 connections and sometimes it can have 3? And how does that work in terms of those valence electrons, if you remember those from physical science? Come back to that in a second. But for now, we're going to make our own key up on the top. This is the amino group. Pause and copy all of this as needed. It's up to you to figure out how you need to do that. And an amino group is a nitrogen atom with 2 or 3 hydrogen atoms. Ooh, I was running out of room there. So an amino group is a nitrogen atom with 2 or 3 hydrogen atoms. That's the amino group. That's one part of an amino acid. The amino group is just one part of the amino acid. Another part is this part right here. This part right here. I'm going to try moving this one over a little bit since I ran out of room on the last one. This is the alkyl group with a side chain. And the R stands for the side chain. The alkyl group has a carbon and a hydrogen and a side chain. So let's go ahead and add that. The alkyl group with a side chain is R, a carbon atom, and a hydrogen atom. One of the things that's going to be important to understand is that R, the side chain, that's variable. It can look like a lot of different things. So I'm going to come back to that as well. But let's go ahead and get our third part here. The third part, of course, is the carboxyl. And that's this part right here. So go ahead and pick a third color for that, and let's go ahead and make our key here. This is the carboxyl group, and the carboxyl group contains a carbon atom and oxygen atom and a hydroxyl. Remember that OH is hydroxyls. You will need to know that for your test, right? So there's that hydroxyl, there's the carbon, there's the oxygen. Notice that this is two lines. This is a double connection. It's connected to the oxygen twice. Carbon has to have 4 connections, so there's 1 connection, 2 connections, and then it doubly connects to the oxygen to get the 4 connections that it needs. Oxygen has to have 2 connections to feel complete and whole, to have that full outer shell of electrons if you had physical science. And so oxygen has to have 2, and so here it doubly binds to the carbon, right? So this is the basic structure of an amino acid, right? And all amino acids have this structure with a variety of different side chains. Pause and copy if you need to. Let's go ahead and look at a couple of examples of these. All right, so we're going to start with kind of this idea. Here is showing there's that carboxyl group, and here is showing here's the amino group. They're using 3 hydrogens this time. And then here is your alkyl group. And there's a variety of different chains that can take place with this. So as we're looking at this, you can see here is that amino group. It's the nitrogen with 3 hydrogens this time. Here is the alkyl group. There's the hydrogen, the carbon, and this is the side chain, and this is the carboxyl. And there really should be a little OH there. They didn't write the H because of, like, sometimes these things like ionic combinations happen and it gets weird, but just kind of for now, we'll keep it with that's an OH group. Hydrogens are just really tricky. They can misbehave sometimes. So sometimes hydrogens rebel and they break free and they leave an empty connection. And that's what happens too with the 2 H's instead of 3. One of the hydrogens kind of rebelled and left an empty connection on the nitrogen. When it leaves an empty connection, that atom is called a radical. And it's not usually going to last very long without connecting to something else, right? But that's why sometimes one of the hydrogens is missing, because it was a rebellious hydrogen and it took off and left an empty connection. Shame on that hydrogen. All right, so here is the amino group. Here's the carboxyl. And this is the alkyl group. The shaded part is the side chain. Here we have, again, an amino group, a carboxyl. Here's the alkyl group, and the shaded box is the side chain. Amino group, carboxyl, alkyl group, and the shaded part is the side chain. So that you can kind of see that this, like, sort of the head structure is always the same, right? There's the amino, the H and the C, carboxyl, the amino group, the H and the C, the carboxyl, amino, H and C, carboxyl, amino, H and C, carboxyl, amino, H and C, carboxyl. It's the side chain that varies. And that side chain that can vary is why there are so many different types of amino acids. And so there's a few of them shown here. And you can see that these are still organic molecules composed mostly of carbon, hydrogen, and oxygen. And there's a few extra things in there. There's some nitrogen. Some of them will have some sulfur. Yeah, histidine is an interesting one. If you take an antihistamine for allergies, you're trying to block that amino acid in particular. It's related to the immune system. Here are kind of a bunch more examples. And again, you can see all of them have this amino group. All of them have the carboxyl, perhaps with a rebellious hydrogen. And then all of them have the alkyl group that has the hydrogen, the carbon, and some sort of a side chain, right? And there's a variety of different examples of these. Maybe some of them you've heard of. Tryptophan is what's in turkey at Thanksgiving dinners that makes you sleepy if you eat too much of it. Isoleucine is involved in helping your red blood cells to work properly. So there's like a lot of different ones. We'll see methionine come up a few times. That's always the end of a chain. And so there's like a lot of different amino acids. They have different side chains, but they all have the amino group, the alkyl CH, and the carboxyl group with possibly a rebellious hydrogen. To kind of look at some of these just as some examples, because I did create some examples for you guys. Here is this one right here. This one right here, you can see the blue is added. We haven't seen blue yet. Blue represents nitrogen. The blue represents nitrogen, so right off the bat you can tell then that this is our amino group, because the amino group is the nitrogen with either 2 or 3 hydrogens. In this case I gave it all of its hydrogens. I did not let it have a rebellious hydrogen. Over here, this is our carboxyl group. And you can see there's the carbon, there's the doubly bonded oxygen there, and here is a hydroxyl. Carbon and hydroxyl makes carboxyl. That's usually when they're referring to carboxyls, what they're looking at. They don't kind of include him in the name as much. A carbon and a hydroxyl makes a carboxyl. So there's my carboxyl, here's my amino group. And then this is my alkyl group. And you can kind of see that there's the tiny little hydrogen and the carbon. And this is the side chain for this particular amino acid. This particular amino acid is isoleucine. And so isoleucine was the one that I mentioned that it helps your red blood cells to work. It helps you to have good energy levels. It also helps to control your blood sugar. So isoleucine is one that is an amino acid that works with your circulatory system, with your blood. It is what is called an essential amino acid. So some amino acids you can get from your diet, and only from your diet. And then some amino acids, your body can actually make them. If it's one that your body can make, it's called non-essential because you don't need to get it from your diet. Your body can make it on its own. This one is an essential amino acid because your body cannot make this, but you need it for your blood to function properly, so you have to get it through your diet. It's an essential amino acid because you have to get it through your diet. And if you are not getting enough of it through your diet, it can affect your energy levels and a variety of different things. So this is another one of those reasons that people talk about eating a nice, healthy, balanced diet. We've looked at that with carbs. We've looked at that with lipids. But even with amino acids, this is important. This is one of the building blocks for proteins that help your blood, and you cannot make it, so you have to get it from your food, right? So that's what essential amino acid means. I've got another amino acid here, And again, you can see here's the amino group. This one's only got 2, so he does have a rebellious hydrogen in there. This one right here is my carboxyl group with the carbon and the hydroxyl, right? And then here is my alkyl group with the carbon and the hydrogen. This one has a very small side chain. This one's a very short side chain in particular. And this right here is called alanine. Alanine is a non-essential amino acid. Basically, your body can make it. You don't have to get it through your diet. Alanine is found in food, particularly in meats, but you don't have to eat it because your body can make it. There seems to be some links between alanine and a glucose cycle in your blood that affects your blood sugar levels. And so people are finding that problems with this amino acid can be linked to type 2 diabetes. So there's another example. And again, you can see there's the carboxyl, there's the amino group, and here's the alkyl group with a very short side chain in this particular case. Here is another example. This one I like is kind of fun. It has this fancy fun shape, right? You can still see here's your amino group. This one right here has 2 hydrogens, so he does have a rebellious missing hydrogen going, but there's my amino group. Here is my carboxyl group. It's kind of getting crowded out by the other one. Here's my carboxyl group with that doubly bonded oxygen and the carbon and the hydroxyl. Remember, hydroxyl is a hydrogen and an oxygen. So there's my carboxyl. And then I've got my alkyl group, the carbon with the hydrogen, and this is a really interesting side chain going on this one right here. It makes this hexagon shape. Let's see if we can do this to kind of get this one to behave. So this one right here is called phenylalanine. Maybe you've seen on the sides of like soda cans or something, phenylketonurics, beware, this contains phenylalanine. Because there's an enzyme that digests this amino acid that if you don't have it, you're not able to digest this amino acid. It's interesting though, because this is an amino acid that our body does use. It is an essential amino acid, means that you have to get it from diet. Your body does not make it. And they've found that problems with this particular amino acid, if you're not getting enough, it's related somehow to depression, to skin disorders, and even to the experience of pain and how you experience pain. And so this is another one that if you want to avoid those things, you have to get it through your diet because your body does not make this. I don't know what that means for phenylketonurics that are not able to digest this so they can't eat it. Do they struggle with depression and skin disorders? I'm not really sure. I've never researched that disease called phenylketonuria. But your body is not going to make this one, so it's an essential amino acid. You have to get it through your diet because it is part of maintaining your health, right? So there's one. So far, all of the side chains that we've looked at have been composed entirely of carbon and hydrogen, but some side chains have other elements, like this one, for example. So this one right here is called glutamine. Put it sideways so he'll fit better. Here's my amino group. There's the nitrogen with the 3 hydrogens. He has no rebellious hydrogens. Here's my carboxyl with my carbon and my hydroxyl, hydrogen and oxygen. Here's the alkyl group with his carbon and his tiny little hydrogen attached, right? And here is the side chain. This side chain has nitrogen in it, and side chains might have nitrogen in them, they might have sulfur in them. Some of them are just hydrocarbon side chains. This one has some oxygen as well. This one right here, glutamine, is another one that is a non-essential amino acid. That means that your body does make this one, so you do not have to get it through your diet. It is active and plays a role in your intestines, keeping your intestines healthy, and even just it's a big part of your immune system. It's one of the molecules that your immune system uses quite a bit. So periodically, if you are experiencing an injury or an illness, you're going to need extra glutamine. And so even though it is non-essential, you can make it, your body does make it. There are times where you might need more than your body can make. And so then you'll have to get it from diet. It is in a variety of foods, both plants and meat. And so if you are injured or sick, you're going to need to eat a little bit of extra of this to help you to recover, because your body does make it, but it might not make enough when your immune system is having to work a little extra hard. So those are just some examples of some amino acids. And again, what they all have in common is an amino group, a carboxyl with this little hydroxyl, and then the alkyl group with the carbon and the hydrogen and some sort of a side chain. And there's lots of different kinds of these, right? All right. Here we go. So next it's going to get into, if proteins are chains of amino acids, how do these amino acids connect? We looked at how carbohydrates can connect when we looked at the monosaccharides, and one of them loses a hydroxyl and one loses a hydrogen, and they connect at that point. And then the pieces that were lost form water, right? And you can make big, long chains of carbohydrates. Well, amino acids can also make big, long chains. So let's look at how it happens. Here we go. In the synthesis of a peptide, remember that a peptide is a chain of amino acids. The hydroxyl group of the carboxyl of one amino acid and the hydrogen of the amino group of the next amino acid are enzymatically removed. Notice this is the same idea that we saw with forming a chain of sugars. One of them loses a hydroxyl, one of them loses a hydrogen, and then they are going to connect at that point, right? So here we go. And the synthesis means making. Synthesis means to make something. In the synthesis of a peptide, which is a chain of amino acids, the hydroxyl group of, and it's from the carboxyl, right? That's where the hydroxyl is, of the carboxyl group of one amino acid, and the hydrogen of the amino group of the next amino acid are enzymatically removed. In particular, it's the hydrogen from the amino group. So the carboxyl loses its hydroxyl, and the amino group loses a hydrogen, right? And that's kind of the beginnings. So the nitrogen from one amino acid bonds with the carboxyl carbon of the adjacent amino acid, and this bond is called a peptide bond. We now have a dipeptide. Remember that di is a prefix meaning two. So a dipeptide is two amino acids. It is the smallest protein. A protein, an amino acid by itself is not a protein. It has to be connected to at least one other amino acid to be called a protein. So once you have two connected amino acids, you have a dipeptide, and that is the smallest protein that exists. And just like we saw with the carbohydrate chains, in the course of the formation of the peptide bond, a molecule of water is given off. We lost a hydroxyl and a hydrogen. They're going to combine to form water. All right, so we're going to flip this over and take a look at what this looks like on the other side and how this works. And I'll do this with my molecules as well. And again, pause as you need to do the highlighting and the notes and all of that kind of stuff like that, right? All right, so here we go. Here we have two different amino acids. It's not showing us what the side chain is. R is just generic side chain. It could be anything. So I don't know what these two amino acids are, but it is going to connect them, right? And so what has to happen is one of them is going to lose a hydroxyl from its carboxyl, and one of them is going to lose a hydrogen from its amino group, and then they're going to connect at that point. Once the carboxyl loses its hydrogen, it just has a C. Once the once the carboxyl loses its hydroxyl, it just has a C. And once the amino group loses its hydrogen, it just has an N. And so that's where they connect, right? So it's going to lose this and this. Those are going to come off to form a water. And that leaves another one of those gaps. And aside from the rebellious hydrogens that don't want to cooperate, they don't like having unconnected spots, right? They want their minimum number of connections, which again, if you had physical science, has to do with the valence electrons. So carbon suddenly is missing a connection. Nitrogen suddenly is missing a connection. So they're going to connect to each other, right? And because the hydrogen and oxygen and, well, the hydroxyl and the hydrogen also are missing a connection, they connect to each other to form the water molecule, right? And so if we're looking at our little models, here's how we do this. Here is one amino acid. You can chain's kind of off the edge, but that's okay, it'll still work. And here is another amino acid with a really tiny chain, so that one you can see nice and easy, right? And we said that basically one of the amino acids, and it doesn't matter which one, is going to lose its hydroxyl from the carboxyl group. So here's my carboxyl group. My carboxyl just lost its hydroxyl, right? The other one is going to lose a hydrogen from its amino group. So here's my amino group, and he just lost a hydrogen. This does leave us with a connection, right, that is missing. You can see there's a hole there, right? He's missing a connection too. And they don't like to be missing a connection, so they will connect to each other. And these guys, they don't like missing connections either. And so they connect, and there's our little water molecule. And this right here is officially a dipeptide, right? And that means this is one of the smallest proteins that I could make. And I could connect all of my little samples in the same way and make a big long chain. Again, if I had enough pieces, I could make a chain long enough to wrap around my entire room. And that would be a polypeptide because it has a lot of amino acids. But this one right here is a dipeptide because it has 2 amino acids. So again, just as a recap, one of them, the carboxyl group, loses its hydroxyl. One of them, the amino group, loses a hydrogen. Those lost pieces are going to become a water molecule, and the 2 atoms are going to connect where the pieces were lost, right? The 2 amino acids are going to connect where the pieces were lost. So that's another 4-step process. You probably should pause this video, rewind it, and take some notes on that 4-step process for the test. So I'll review it one more time. One amino acid loses a hydroxyl from the carboxyl group. One amino acid loses a hydrogen from the amino group. The hydrogen and the hydroxyl that were lost will connect to form water. And the 2 amino acids that just lost their pieces are going to connect to each other in that place where the pieces were lost, a carbon to a nitrogen. That's how you make a protein, by connecting amino acids. Let's go ahead and continue on. Okay. Says, notice that the far right side of the dipeptide, it could join with the carboxyl group of an adjacent amino acid, and the far left of the peptide where it could also be extended. As we're looking at this, right, and what they're saying is, here's my dipeptide. There's another carboxyl over here that could lose a hydroxyl and connect to something else. There's another amino group over here that could lose a hydrogen and connect to something else. And again, we can keep building that chain longer and longer and longer to make a polypeptide. So that's what this one is kind of saying. Far right side of the dipeptide could join with the carboxyl of another amino acid, and the far left could join as well and be extended to the amino group. And additional amino acids are added to the growing chain of peptides. And it'll just keep being added and added until the final amino acid has been added and you get a polypeptide. And so ribosomes are the organelle that builds proteins, and what a ribosome does is connect a bunch of amino acids. And that ribosome is going to keep connecting amino acids and keep connecting amino acids and keep connecting amino acids until the nucleus gives it the instructions to stop. And the nucleus also gave the ribosome the instructions for how to connect these amino acids, like what order, which ones, and all of that. So amino acids form a chain, and it's the ribosome that creates that chain using the instructions from the nucleus, right? And at some point, the nucleus will give a stop code to the ribosome, and the ribosome knows that that's the end of this chain of amino acids then, and it releases that chain and will start making the next chain. Whatever the nucleus tells it to make, it's going to make the next chains. So this is how ribosomes make proteins. Inside of the ribosome, it's making all of those connections for all of those amino acids. So, goes on to say here that the order and the number of amino acids in the peptide chain is determined by the cell's genes, which are, of course, in the DNA. And then once the peptide has been formed, there are some more changes that need to be made. Additional modifications occur. And so like the ribosome will build the chain, and then it's a linear peptide. It's just this big long chain, but it's not quite done yet. It needs to be shaped, and they have a variety of different shapes, and the ways that they build their shape involves polar and nonpolar pieces. Some of the amino acids are polar, some are nonpolar, so they'll attract or they repel each other. But they need to be shaped. So that's kind of where this is going to continue from here. Wish I could get this to focus better. Sorry guys though, it just doesn't seem to want to focus better. Ooh, hey, maybe that is better. Nice. All right, so as amino acids are added to the growing peptide, again, the ribosome does that, right? So let's say by a ribosome, a polypeptide results. And when the polypeptide is modified to its working structure, it is called a protein. So when it's just a chain of amino acids, they call it a peptide or a polypeptide. It's not technically called a protein until it reaches its final shape. When the polypeptide is modified to its working structure, it's called a protein. And basically that's saying when it's given its shape, that's when they officially call it a protein instead of a peptide chain, right? So you got your ribosomes going to make proteins, but really it's making peptide chains. It's connecting all of them, right? And then that peptide chain has to be shaped, and once it's shaped, it'll be called a protein. And like as you look at the back, here is the linear peptide, right? Let's go ahead and write linear peptide here. And the linear peptide has a variety of different amino acids. So this is histidine, tyrosine, lysosine, leucine, histidine, valine. The ribosome is just following the instructions. These can be put together in like hundreds, if not thousands of different orders, and so the nucleus is what's got the DNA. That's the instructions for how they put together. The ribosome is just following the instructions and says, okay, I need a valine here. What's connected to the valine? Oh, okay, I need a histidine next, right? And so that's kind of where they get their instructions from, is the nucleus, right? We have this right here is what we just learned is a peptide bond, right? This is where the carboxyl of this one is attached to the amino group of this one because of the lost hydroxyl and hydrogen, right? And so this peptide bond is this right there, right? That's a peptide bond. And then these are the amino acids. And there's lots of different ones, each with a different side chain. And so when they connect, we get a linear protein, right? But a linear protein is a linear peptide, not called a protein yet, technically, because it's not shaped yet. So if you're a biochemist, you wouldn't call it a protein yet. You'd call it a peptide, a linear peptide, a peptide chain, right? And then as you look at the rest of the slide, you can start to see some of the shapes. Some of them are spiraled. Some of them are just like these globs. Some of them are multiple globs all kind of connected together. So we'll read about these in a minute. But so far, you've got, this one was a dipeptide because it was two aminos attached. We looked at how they attach by losing a carboxyl and a hydrogen, which then combine form water. That attached spot is called a peptide bond. And in a linear peptide, there's going to be a bunch of amino acids that are bound together with those peptide bonds, right? So that's kind of where we're at so far. Let's go ahead and kind of continue on. Starting right here. The linear sequence of amino acids in a protein is referred to as the protein's primary structure. When it first comes out of the ribosome, this is what it looks like, right? So that's why it's called the primary structure. That's what it looks like first, fresh out of a ribosome. It says in the diagram entitled primary structure, they showed 6 amino acids. That was the leucine and the tyrosine and all of those linked together by the peptide bonds. We're not going to read the coloring instructions. It does give the names of the amino acids that are in that chain if you wanted to see it. So the VAL was valine, the LEU was leucine, the LYS was lysine. I'm not going to have you memorize all of the amino acids. There's like tons of them, and oh my goodness, they can combine, right? What you do need to know is that this is the primary structure. The linear protein is the primary structure when it first comes out of the ribosome, right? We have a secondary structure of protein, refers to the folding and twisting patterns of the protein chain. So it's going to get folded and twisted. One pattern that proteins can assume is a helix. Notice though that that's just one pattern that, or one shape that they can take, right? And so that's kind of what it was showing here. Fresh out of the ribosome, we have a linear protein, and the secondary structure is that it can be twisted. One example is it can be twisted. There's some other ways that it can go too, but in this example, the secondary structure is it gets twisted, right? So that's that. Helix is one type or example of a secondary structure, right? And maybe it'll stop there, maybe not. Continuing on, certain polypeptide chains can fold back in on themselves to form folded proteins, right? So some of them will stop at whatever is their secondary shape, which might be a helix and might not. Oh, pausing for a second, guys, sorry. Sorry about that, guys. Okay, let's continue on. So some of them, they'll stop at their secondary shape, which may be a helix, but some of them will fold back on themselves, and then it's called a folded protein. It says these proteins are globular proteins. If I take something in like, you know, it's this nice neat shape and I fold it all up and I make it's a glob is a shape, right? It's just this big messy ball. And so these are called globular proteins because they just get all folded into big messy balls. And so that's kind of, I mean, it shows their three-dimensional shape. Let's go ahead and take a look at an example of this. So they're using this to show the spiral, right? This spiral is this right here, right? Notice that this spirally thing, imagine a slinky. Imagine the slinky getting all folded up in half, and like you can see that it folds up this way, it folds there, it folds there, it folds there, it kind of shoots out the back side. And so this spiral can get all folded and into like halves and pieces and whatever, and then it just becomes this pile. This is called a globular protein, referring to the fact that its shape is just this glob, which is this messy pile, right? Because that twisted helix just got all folded on top of itself. And so it's a globular protein, also known as, AKA means also known as, it's another name for a folded protein, right? Because it's been all folded up on itself to make a glob. That's where the name comes from, right? So let's go ahead and kind of continue back on this side. So says the protein in the figure, the one that they're showing in particular, is one of 4 polypeptides in a hemoglobin molecule of a human blood cell. So that particular protein is part of a red blood cell. And the hemoglobin molecule in the blood cell, it's called a heme group. And so that's just kind of giving you an example. So this one right here, that was this spirally helix shape that then got all folded up on itself. You can see where it gets all folded up. Woo. That is a folded protein, a globular protein, and in particular, this example is one that would be found in a red blood cell, right? And this is one heme group. And where it's going to go with this is that these can combine with other globular proteins, and they get stuck to each other, right? So here's one heme group, here's a second heme group, here's a third heme group, and a fourth heme group. And so this would be kind of that fourth structure. Quaternary means fourth. Is when you take several of these and you combine them into an even bigger glob. Let's go ahead and look at that next. So the final structure we show is the quaternary structure, in which multiple polypeptides are organized together. Again, we took one folded protein and combined it with several other folded proteins. They all started as a linear peptide that got spiraled to make a helix. The helix, sort of like a slinky, got all folded up into a pile called a globular protein or a folded protein. Now we're combining multiple folded proteins to make this quaternary structure. In this particular case, this is got 4 chains. They label them H1 through H4. And each one is different, but one of them is identical to the folded protein in the third. So there were 4 different folded proteins, but one of them looks like the previous diagram is all that same. This final structure of the hemoglobin protein is found in red blood cells. It's important for transporting oxygen throughout your body. And so essentially this right here is that one. These are different. They're not exactly the same, but they all combine together to make a superglobular protein, right? And so this is, whoops, 2, that says 2, just roll with that. 2 or more folded proteins combined. And some of them will stop here, just like some of them stop here, right? Some of them stop here, but some of them, they start forming all of these combinations, right? So we kind of go from a linear protein that gets shaped, maybe to a helix. The helix gets folded all over itself to make a globular protein or a folded protein. And then the folded proteins can combine to make an even bigger globular protein. And that's kind of where we're at today. If you have questions, go ahead and email me. Hopefully you were able to pause and get all the notes. If not, you can obviously rewatch the sections that you missed. Reminder, you have a test coming up later on this week. And so watch for optional Zoom for questions and then required Zoom for the test. See you guys soon.